Hello, and welcome everyone to Bio-Rad's webinar series on Droplet Digital PCR. This is our 8th webinar in the series Bio-Rad is hosting on ddPCR. My name is Mohini Patil, and I'm the Global Product Manager for Droplet Digital PCR at Bio-Rad and your host for today's webinar. First, let's discuss the webinar logistics. The webinar will last approximately 1 hour. Dr. Maar will present on tips and tricks in ddPCR assay design. Following the presentation, we will have live questions and answers with Dianna. Throughout the webinar, feel free to use the Q&A box on the left-hand side of your screen in order to submit your ddPCR questions directly to us. We will get to as many of your questions as we have time for. However, if we cannot get to your question today, we will personally follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. Now, a little bit about our speaker, Dr. Dianna Maar. Dr. Maar studied at the University of California, Los Angeles, where she received both her bachelor's and doctoral degrees, majoring in microbiology, immunology, and molecular genetics. During her graduate training, Dianna studied antibiotic resistance, ribosomal mechanisms, and protein expression. She continued to work on translational and protein expression at the Scripps Research Institute. She expanded her research interest at Sandia National Laboratories, where she worked on developing biofuels from protein fractions, identified virus entry mechanisms, and gene expression profiles for Nipah virus. As a senior scientist at Bio-Rad's Digital Biology Center, Dianna is part of several teams where she is working to develop new tools and applications for Droplet Digital PCR. The title of Dr. Maar's talk this morning is Droplet Digital PCR, Tips and Tricks for Assay Design. Without further ado, let's get started. Dianna?
Thanks a lot for your introduction, Mohini. Before we get started, I want to make everyone aware that Bio-Rad has recently launched a line of PrimePCR assays that are wet lab validated for ddPCR in both the QX100 and QX200 systems. Currently, there are over 500 assays for copy number and rare mutation detection. Also, the PrimePCR gene expression assays for EvaGreen have been approved for use in ddPCR with greater than a 90% success rate. Please see the link provided for the current list as it's always growing. Next, I will give a brief overview of the technology, benefits, and workflow for digital PCR, or ddPCR. Here, I am showing two different ways that you can quantitate your nucleic acid materials. In the first example on the left is qPCR. All of the components for the PCR reaction reside in a single tube, and fluorescence is measured after every cycle of the reaction. And the nucleic acids use a standard curve or dilution series of some known quantity. In the tube on the right is Droplet Digital PCR, or ddPCR. Here, the PCR reaction has been partitioned into thousands of uniform-sized droplets. In this method, the fluorescence is measured per droplet after all of the PCR cycles have been completed. This generates an absolute measurement that is based on the actual amount of nucleic acid material present in the sample. This has many benefits. Data is absolute with no standard curve needed. It allows you to compare data from day to day and lab to lab, and thousands of droplet partitions result in high precision measurements with very small error bars. And finally, the QX200 allows you to use EvaGreen-based assays as well as probe-based TaqMan assays. This is a diagram of the QX200 system and its workflow. The QX systems have many advantages, but throughput and workflow are two of the biggest, followed closely by cost per sample. You can affordably run 96 reactions, each having 20,000 partitions made. Cost per sample is comparable to qPCR, and without the need for a standard curve or triplicates, it can be a cost savings. After making the reaction mix, the workflow is very simple and intuitive, shown here. Since the ddPCR reaction is read at endpoint after thermal cycling is complete, each droplet that has fluorescent signal can be counted as a positive. This results in more flexible assay design rules as well as the ability to see how well the assay performs. As I mentioned earlier, our QX200 platform supports both TaqMan detection and EvaGreen detection. Here is a simplified diagram of how a TaqMan assay works. TaqDNA polymerase uses the primer DNA complex to know where to begin polymerizing. The Taq polymerase enzyme has 5' to 3' exonuclease activity that will cleave the probe as it polymerizes the new strand. This probe cleaving is necessary for fluorescence because it frees the 5' fluorophore from the probe's 3' quencher. The fluorophore is shown as a reporter in the above example. TaqDNA polymerase is also very thermostable and has a large range of temperatures that it can both tolerate as well as perform polymerization of new DNA. This is necessary since high temperatures are needed to melt double-stranded DNA after each round of polymerization. For designing assays, it is useful to know the range of temperatures that the enzyme can tolerate and polymerize efficiently at. The optimal temperature for the polymerization or anneal extend step is 55 to 65 degrees. However, if necessary, it may be possible in ddPCR to go as low as 50 degrees and as high as 75 degrees in some assays. Going below this or above this temperature during the elongation or extension portion of the PCR reaction is not recommended and will likely result in a significant loss of activity and thus the number of total amplicons made. Please note, however, that our one-step ddPCR supermix requires a smaller temperature range, as we will discuss more later. Balancing temperature, assay concentration, and primer probe length are important when designing an assay because in an enzymatic reaction, the components are dynamic, coming on and off the DNA. Since all components have an on and off rate, keeping the amplicon length short increases the number of polymerization events that will finish and thus make a complete amplicon without the polymerase falling off. Depending on the target and the question you are trying to answer, it may be worth trading efficiency for a longer amplicon. Other trade-offs may be necessary as well, such as having a longer primer and or modifying the thermal cycling conditions. We will discuss both the amplicon length and thermal cycling conditions in detail. There are two general assay design regimes shown here. On the left is a common discrimination assay showing a single set of primers used to amplify two nearly identical sequences. In this type of design, two different probes are used to discriminate one amplicon from the other. This type of assay can be used to discriminate single nucleotide polymorphisms, rare mutation discrimination, or deletion insertions in the gene. On the right, the common multiplex assay is diagrammed showing two or more unique targets amplified using two or more primer sets, as well as a unique probe for each. This assay type is typically used for copy number variation, gene expression, pathogen detection, environmental monitoring, and linkage analysis. Here is what the data will look like for a well-performing duplex assay. The 2D or two-dimensional droplet plot in Quantisoft is a plot of the individual SAM-positive droplets in blue, the hex or Vic-positive droplets in green, the double-positive droplets in orange, as well as the negative droplets in gray. This plot can provide a lot of detailed information about your assay's performance. I will show examples of this throughout the presentation. This is a 2D plot of a discrimination assay where two nearly identical targets use the same primers but two different probes for discrimination. This is what the plot should look like. The double positive populations will angle inward due to sharing of the same primers and will result in a slightly depressed double population. In this example, the amplicon differs only by a single nucleotide under the probe region and is a common assay type for detecting activating mutations in cancer. Specificity is the most important aspect of this type of assay and differs from single nucleotide polymorphism assays by requiring high sensitivity and low false positive rates. Next, let's discuss the EvaGreen assay. As I mentioned earlier, the QX200 supports both TaqMan and EvaGreen detection. Double-stranded DNA binding dyes, like EvaGreen, can be used to quantitate DNA using a simple set of primers. Potentially any DNA polymerase can be used for this type of detection, and fluorescence comes from the amount of Amplicon made and the Amplicon length. Benefits include new applications such as detecting longer Amplicons, shorter Amplicons, breakpoint mutations, and splice variant quantitation. Generally, longer Amplicons have higher fluorescence due to having more dye bound per Amplicon, and shorter Amplicons have lower fluorescence due to having less dye bound per Amplicon. This has important benefits with ddPCR and will be discussed in more detail later. Let's take a moment to answer this poll question. What assays are you currently using for ddPCR? Choose as many as apply. All right. So it looks like a lot of you are using, it looks like a lot of people here are using pre-designed assays from commercial vendors. I assume that's for qPCR, and some of that's from ddPCR. So it looks like, yeah. Okay, great. And some of you are designing your own assays, so that's good. All right, thank you. So next, let's talk about efficiency and specificity. These are two common terms used to discuss assay design and optimization that I will be using a lot throughout this presentation. These are my definitions. Because digital PCR reads the reaction at endpoint, efficiency means total number of amplicons made after all PCR cycles are complete. This definition is different from when it is used to discuss qPCR assays because qPCR reads the number of amplicons made per cycle. Although having good efficiency is always desired for any type of PCR, endpoint detection in droplets can be more forgiving of low efficiency or fewer total amplicons made, since a positive droplet can be counted whenever it is distinct or separated from the negative droplets. This can also be true when encountering sample-specific PCR inhibitors. One of the most important things you can do to ensure the best efficiency for your assay is to digest outside the amplicon with a restriction enzyme. I will show examples as well as provide protocols at the end of this presentation. Specificity is also important and can be verified first using bioinformatics and finally empirically using a temperature gradient. General recommendations for ddPCR assay designs are as followed. Primer probe length can range from 15 to 35 nucleotides and is optimal at 17 to 25 nucleotides. Primer and probe melting temperature can range from 50 to 70 degrees and is optimal at 55 to 65 degrees with a 1 to 3 degree warmer probe. Importantly, the probe sequence cannot begin with a 5' G. This will result in fluorophore quenching. Amplicon length can be between 50 and 250 plus nucleotides and is optimal at 60 to 100 nucleotides. Again, remember that shorter length amplicons improve quantification from fragmented or damaged DNA samples. Single nucleotide discrimination assays should have a TM around 55 degrees, have a short amplicon length of less than 90 base pairs, and be designed with a short probe in order to decrease cross-reactivity. Copy number variation assays benefit from matching unknown and reference amplicon lengths, especially with fragmented DNA. ddPCR assay design can be flexible, and many rules can be and sometimes must be traded or broken in order to optimize for the target of interest. A wide range of primer and probe lengths can be used. There's no need to have a G or C on the primer ends or to have the probe 10 degrees higher in TM than the primers. With that said, let me add that if this convenient, familiar rule from these two suggestions can provide a benefit in some cases, then feel free to use them. Also, Amplicon length is one useful way to multiplex in EvaGreen. Let's take another minute here to answer the following poll question. What method do you currently use for selecting assay sequences and calculating melting temperature? Choose one. Wow, that's incredible. It looks like 100% of you are using free software. That's great because we're going to talk a lot about that in the next few slides. Correctly calculating melting temperature is one of the most important factors in assay design. Since melting temperature is a function of oligo-base composition, master mix salt and DNTP concentrations, oligo concentration, and the algorithm used in order to calculate TM, we recommend that you see the Droplet Digital PCR Applications Guide for detailed information on adjusting the salt corrections and thermodynamic settings to Santa Lucia 1998, as well as ensuring that the monovalent and divalent cation settings are optimal. It will also be helpful to take your existing qPCR assays and see what the TM is calculated with these settings. At the bottom of the slide, I have the URL listed for the Droplet Digital PCR Applications Guide. Here are two URLs listed for two free sites that you can use for assay designs, Primer 3 Plus and NCBI Primer Blast. Note that the nomenclature shown here is based on Primer 3 Plus. Next, it will be useful to diagram how to adjust the settings in both of these free sites. Here is a screenshot of Primer 3 Plus. This is the view that you will see when you are in detection mode, which can be selected in the task drop-down menu. You will also need to go to the General Settings tab, circled here, to change the settings to the ones recommended for the primers, and then do the same for the Internal Oligo tab if you are designing a TaqMan assay. If you are interested in checking an existing assay, you can change the task to Primer Check mode shown here. You will also need to go to General Settings and Internal Oligo tabs to change the settings to the ones recommended for ddPCR. In the NCBI Primer Blast site, you must select Advanced Parameters in order to see and adjust the settings. This can be found after scrolling down to the bottom of the first page. Using the settings I have recommended here in a software will get you close to the actual TM of the primer and probe. However, modifications to your assay made during the ordering process can change the actual TM. This is why we always recommend first running a temperature gradient on your assays before choosing an anneal extend temperature. Uncommon assay modifications are made specifically to change the TM of your assay. Some of these modifications are moieties that are covalently linked to the nucleotides, such as MGBs or minor groove binders. And some are chemical modifications made to the nucleotides. Common assay modifications, such as the fluorophore and quencher, are largely accounted for in the recommended settings. Be aware that when using TM enhancers, if they are not designed correctly, they can decrease specificity and cause you to have primer and probe melting temperatures that no longer match up. It is worth commenting that a common misconception exists that a higher TM design will automatically be more specific. This is only true if this allows you to cause your assay to go from a non-optimal temperature to an optimal temperature, such as with AT-rich regions or for very short amplicons. Again, getting all components of your assay to a common TM, that is in the operating temperature for the polymerase should be the goal. Next, let's go over the temperature gradients. The temperature gradient will verify the optimal anneal extend temperature for your assay. When looking at the temperature gradient of the assay, it is helpful to view the data by looking at it in a one-dimensional plot. This is so that the data from the different temperatures will be side by side, allowing you to easily identify the temperature where the assay has the highest fluorescent amplitude and the lowest amount of rain or mid-amplitude droplets. For multiplexing, it is recommended to use 2 or more assays that all have similar optimal TMs or temperatures. In the case shown here, these 2 assays could be duplexed together and cycled at an anneal extend temperature of 56 degrees. In this next example, I show a temperature gradient that reveals a nonspecific product being detected at the lower temperatures. So it is also useful to run a temperature gradient on your assay to ensure specificity, especially with EvaGreen assay designs, because they do not have the added specificity provided by the probe as with TaqMan assays. Let's now discuss EvaGreen detection and Amplicon length in assay design considerations. EvaGreen assays still benefit from shorter length amplicons in difficult samples and efficiency. However, longer amplicons amplified with good efficiency will give higher separation because more dye molecules will bind. Here is a one-dimensional plot showing 3 different length amplicons for RPP30. These assays all share the same forward primer and have 3 different reverse primers and no probe. The lengths are 200 base pairs, 99 base pairs, and 62 base pairs. When run at the optimal anneal extend temperature, the different length amplicons can be easily identified. This change in fluorescence due to a longer amplicon will continue to increase with amplicon length until the amplicon's length begins to decrease the reaction efficiency or total number of amplicons enough to drop the fluorescent amplitude. This is data showing the effect of amplicon length in EvaGreen for 8 different targets. We recommend amplicon lengths around 60 to 200 base pairs with TaqMan, but you can use amplicon lengths that are 50 to 700 base pairs in EvaGreen. Note the extra 2-minute extension time for longer amplicons. Keep in mind that shorter amplicons are generally more efficient and will therefore be more sensitive to length discrimination. See the following publication for more details. This is a 2013 analytical chemistry publication that provides detailed discussion and data for EvaGreen multiplexing using amplicon length and efficiency. EvaGreen in ddPCR has another benefit that stems from amplicon length discrimination features. This is the ability to see and exclude primer dimers by setting the threshold above the primer dimer population. There is no redesign necessary for this assay since the primer dimers are short enough to appear near the negative cluster. Importantly, primer dimers are easily verified as a primer dimer and not a nonspecific product by running an NTC or no template control of just assay with a master mix and water. Let's pause here again and take another poll. What type of assays are you interested in designing? Choose as many as apply. Okay, yeah, thank you. This looks interesting here. It looks like copy number variation is pretty even with single nucleotide discrimination and gene expression. We also have some of you developing pathogen detection or quantitation assays. Great, thank you. Now that we have covered the assay design details, let's take a minute to review the basic recommendations for assay concentration, digestion, and thermal cycling conditions. Always remember the basics and start with our recommended assay concentrations and thermal cycling conditions, as well as a digestion of the DNA with an enzyme that only cuts outside of the Amplicon. If it is desirable to forego the digestion step, please do a concentration gradient with and without digestion to ensure that your quantitation is comparable before skipping this step. Likewise, for making changes to other recommendations. Here is a link to the product insert for the Droplet Digital PCR Supermix, as well as a link to the applications guide for more details. Now that you have tested your assay using the basic recommendations, it may be that you will want to or need to make adjustments to your thermocycling conditions. In order to do that with the best results, it is necessary to understand each step of the reaction and what it does. Step 1 is the enzyme hot start. Step 2 is the denaturing step where the two strands separate and secondary structure is unfolded. Step 3 is the anneal and extend step where the primers are at a temperature where they will bind to the DNA and the polymerase is at a temperature where it can polymerize. Step 4 inactivates the enzyme after all of the cycles are complete. And step 5 is for storing the reaction until you are ready to read the droplets. Note that this is the protocol for the Droplet PCR Supermix and the ddPCR Supermix for probes. See the insert for the thermal cycling conditions and recommendations for the one-step Supermix kit. Next, I will discuss the EvaGreen thermal cycling conditions. EvaGreen thermal cycling conditions are similar to the TaqMan-based conditions, with a few important changes. The most important differences are steps 4 and 5, which are essential for stabilizing the EvaGreen dye. This step will prevent the baseline fluorescence from moving up over time across the plate during reading of the droplets. This will not affect the concentration calculations, but can provide better visual well-to-well comparisons. Next, let's review a little and discuss DNA digestion. Getting the correct TM calculation for matching primers and probe is essential for success. Shorter amplicons exhibit better efficiency. Use a temperature gradient for all new assays. And finally, remember to use a restriction enzyme to digest the DNA. Note that digestion cannot be used for RNA. We have made the digestion step easy by making it possible to add the enzyme directly to the reaction instead of requiring a pre-digestion. Here is the protocol. You can get optimal results by adding as little as 2 to 5 units of enzyme per 20 microliter reaction, with no incubation time necessary. See the product insert for the Supermix for these instructions. We have validated this in-mix digestion with the following 5 restriction enzymes. I have pulled out 3 examples to demonstrate the usefulness of this step. The example shown here is a plasmid sample and can be the most extreme example of the benefits of digesting your sample for primer probes accessibility. Note that not only does the mid-amplitude droplets disappear, but the concentration increases significantly when digested. Plasmids typically have the most secondary structure. Digesting the DNA outside of the Amplicon with a restriction enzyme can rid the region of secondary structure and make the target DNA fully accessible to the primers and probes. This is true for all PCR and is not specific to ddPCR. However, partitioning the sample, as with ddPCR, has made these considerations more obvious and easier to see. Other things that will provide a well-designed assay include having both primers designed with the same TM and performing the PCR reaction at an empirically tested anneal extend temperature. Next, I will show you 2 more examples of how digesting the DNA will offer you the efficiency that you need to get absolute quantitation. It is important that you look both at the amount of mid-amplitude droplets, or rain, as well as the concentrations between the digested and undigested sample, since the rain can be fairly minimal in some cases, yet still result in poor quantitation. In rare instances, you may need to try a few different restriction enzymes in order to find one that will work for a more finicky assay. In this example, this rare somatic mutation assay needed to be tested with 2 different enzymes before one was found that improved efficiency. This is not usually necessary, but can be for some challenging targets. If you look closely here, you will see that digestion was required in order to obtain distinct clusters, correct concentrations, and good fractional abundance. Note that damaged DNA, such as FFPE extracts, will cause rain or mid-amplitude droplets even on the best performing assays. This is why we recommend using good quality control DNA for validating an assay. The next example is a duplex assay used for copy number variation. In this example, it is less obvious that digestion is needed because both assays have minimal rain. However, the copy number went from 1.73 to the expected number of 2 by simply adding MSC1 enzyme directly to the supermix. If the sample DNA is fragmented and or chemically damaged, it is wise to keep the Amplicon length short, as this will be able to amplify and count more of the DNA present. In the case of copy number and other ratiometric comparisons, they are also best done using matching Amplicon lengths for all of the assays used. So far, we have covered the assay design, the digestion of the sample, and now we will cover more suggestions for modifying the thermal cycling conditions. Different thermal cycling protocols have been developed for qPCR with names such as the 3-step protocol or a step-up protocol, and these same modifications can be made in ddPCR thermal cycling. In addition to these, ddPCR is unique in that you can also use split cycles by having the first 10 cycles at a lower temperature and the last 30 cycles at a higher temperature, and vice versa. This is because you read the sample at the end of the PCR reaction and not after each cycle. When making these adjustments, remember to use at minimum the times shown here in our recommended protocol. And although I show the TaqMan thermal cycling protocol, similar adjustments can be made in the EvaGreen protocol. The anneal extend time can be lengthened in both EvaGreen and TaqMan runs. This is usually done when working with a longer Amplicon or with an Amplicon having repetitive sequences or some known secondary structures, etc. Note that it is not recommended to extend the number of cycles beyond 45. It often does not result in any improvements. Finally, changing the denaturing temperature from 94 degrees to 96 degrees for 30 seconds can be done, but is recommended as a last adjustment that can be made after digesting the sample DNA and running an anneal extend temperature gradient. Often it can eliminate or reduce any lingering rain. With that, I will leave you with a list of some useful URLs, and thank you for your attention.
Dianna, thank you very much for all those great tips. If the audience would like a copy of today's PowerPoint presentation, a copy of the QX200 ddPCR brochure, applications guide, and other resources, they are available to the link at the left of your screen for download. The webinar is also available on demand within 24 hours. If you would like any additional information, please feel free to reach out to your local Bio-Rad Droplet Digital PCR specialist as well. Dianna will now take questions. Our first question is regarding assay design. What adjustments should be made if the Amplicon has a very high GC content?
That's a really good question. The best thing to do with a high GC content Amplicon is to keep the Amplicon short and the assay within the optimal TM range. With that said, if your Amplicon needs to be on the longer side, I've seen longer primers with a higher TM be beneficial for long GC-rich targets. Remember to stay below 70 degrees for the TM of the primers, and be aware that poor block uniformity on an old thermocycler can cause edge effects or poor amplification in wells around the edge of the plate. Note that long GC-rich and or methylated amplicons can be sensitive to too much temperature variation across the block as well. So for best results, calibrate or test your thermocycler and update if necessary.
So our next question is regarding EvaGreen Detection. So in one of your earlier slides, you showed signal strength related to Amplicon length. The background in that slide was also high. Do you know why the background is higher, and is there a way to avoid that?
I assume that you mean the baseline of the negative droplets in that question. So with that, the primer sets that you use will cause variation in where your negative droplets sit. That is something that does not seem to correlate with the amplicon length and will just vary from assay to assay. So as far as we can tell, it's not something that can be accounted for or considered in amplicon length multiplexing.
So our next question is regarding restriction digest. So you mentioned restriction enzymes can be used to digest the DNA. Would you recommend QIAsure to share DNA?
Actually, we don't, because fragmenting your DNA randomly will also randomly fragment your target of interest and can be actually cause you to quantitate much less than is actually present in your sample. So no, we don't recommend that.
So along the same lines on DNA digestion, could you elaborate a little more on the conditions for DNA digestion?
So the conditions are actually very simple. You just simply add the enzyme to the mix, and you don't have to do any incubation. There's no waiting time. There's nothing special that needs to be done or nothing else that needs to be added. However, we do recommend that you use the diluent that is provided by the enzyme provider for any dilutions that you do, and don't store that enzyme in the diluent. So you have to throw away what you don't use from that first day. So just only dilute what you're planning on using.
Our next question is regarding the specificity between the two methods you mentioned, EvaGreen and Probe. Is there a relative difference? And then what is the difference between the two methods for the same primer mix?
I'm sorry, can you repeat that question?
Sure. So the question is regarding the two methods and the specificity of EvaGreen and Probe. Is there a difference between the two methods?
Well, so as I mentioned during my presentation, EvaGreen Detection uses a simple set of primers, and so there's no probe. So the chance that an off-target amplicon could be made and detected is a little bit higher in those cases because the probe sequence adds another level of specificity when you use TaqMan assays. With that said, it is usually possible to, using bioinformatics as well as a temperature gradient, identify any off-target amplicons that might be getting amplified before you begin using the assay.
So early on during your presentation, you showed some 2D plots. One of them showed the 2D plots had the different double positive populations. Could you expand upon that a little more?
I assume that you mean the difference between the discrimination assay and the multiplex assay. In the discrimination assay, you're amplifying with one set of primers and you're detecting with two different probes. And in that case, because they are sharing primer resources, that double positive population will move inward slightly, and that's just a resource that's caused from the primers, actually from them sharing the same set of primers. And so that slight move inward is nothing to worry about. It's simple and easy to threshold still, and shouldn't be a concern, but should be what you expect to see even with the best performing assay.
So we have another question regarding digestion. For RT-PCR, using first-strand cDNA synthesized from RNA, will the restriction enzyme digestion help?
That's an excellent question. Actually, that should not cause any improvements because the cDNA that's made is already going to be in smaller, shorter lengths, so it really does not provide you with any benefit.
So along the same lines, do you need a restriction digestion step for FFPE samples?
That's another very, very excellent question. We've recently done a study on FFPE samples with and without digestion, and we found that most of the time that the digestion step helped even with the most fragmented FFPE samples. So yes, we do recommend that you digest with FFPE samples.
And then regarding FFPE samples, you mentioned that Bio-Rad sells wet lab validated assays. Have these assays been used on FFPE samples?
Yes, we've actually run panels of our assays on FFPE samples, and so they have all been tested on FFPEs.
So our next question is from a user who's been running SNP detection assays, and they have noticed the negative droplet intensity increase down the columns of their 96-well plate. Do you have any suggestions as why this occurs and how that can be minimized?
I'm sorry, can you repeat that?
So we have a user running SNP detection assays, and they have noticed the negative droplet intensities increase down the columns of the 96-well plate. Why does this occur, and do you have any suggestions to minimize this?
So that is actually an interesting observation if that's happening down the column. I can think of two things off the top of my head. One would be to ensure that they're mixing their supermix properly, as you wouldn't want some of the wells to have more assay than other wells, which could change your baseline fluorescence. And the second thing that I can think of is making sure that they use the 98 degrees at the end of the thermal cycling protocol for 10 minutes. So just to ensure that their protocol cycling parameters are as close as possible to the ones that we have recommended.
Our next question is regarding sample prep. What is the best method to isolate DNA from cells for droplet digital PCR use? Does the DNA viscosity cause any issues with droplet formation?
So that's a great question. We do have a limit of about 1 microgram per 20 microliter reaction that we recommend, and that is due to viscosity considerations. However, digesting your DNA with 1 and sometimes even 2 enzymes can actually help with that viscosity and cause the DNA to be added, not to cause any problems at these higher concentrations. However, we do recommend that you stay at or below 1 microgram of DNA per 20 microliter reaction.
So the next question is regarding some results. Absolute quantification of an HIV DNA assay shows a lot of rain. Do you know what causes this, and is it giving the right quantification number?
So that is a really good question. Absolute quantification can be affected by rain, as we discussed. And so viruses are typically high in secondary structures, so it is especially important to make sure that you're using an enzyme that is allowing you to fully amplify your region that you're interested in. So my recommendation would be to test several different enzymes in order to try to get rid of that rain.
So we have another question on restriction digestion. You mentioned that the restriction digestion step does not need any additional incubation time. will the restriction enzyme be inactivated with the first 95-degree step of the thermocycling protocol?
Yes, absolutely. That step will absolutely inactivate the enzyme. Good question.
Coming back to FFP samples, do you have any recommendation for DNA repair kits?
We are currently investigating that, and we should have an applications note out for you on that. So just keep checking our website for details.
So our last question is regarding EvaGreen. You mentioned that you can multiplex using EvaGreen. Could you reiterate on what considerations need to be kept in mind while multiplexing with EvaGreen?
Yeah, so that's a great question. We have the publication that I showed earlier in the presentation, and that goes into a lot of really good details. But some of the ways that were used in that paper were using both a TaqMan assay combined with EvaGreen primers. So in one assay there was a probe, and in the other assay there was no probe, and so that's one way that you can use the EvaGreen for multiplexing. You can also do length, so adding an Amplicon with about 30 nucleotide different lengths can give you good discrimination between the two targets. You can also use temperature. So we talked a lot about how temperature can affect the number of Amplicons made, and so if you wanted to in EvaGreen, you could have one primer set designed to a lower temperature and one primer set designed to a higher temperature, and then you could run the reaction at the temperature that is higher and then see two distinct clusters, one from the Amplicon that is designed with primers that are for the lower temperatures.
Thank you, Diana, and thank you everyone for attending the Bio-Rad Droplet Digital PCR webinar today. As a reminder, today's webinar will also be available on demand within the next 24 hours. For those of you who had questions we are not able to get to today, someone from Bio-Rad will follow up with you in the next few weeks. For more information on Droplet Digital PCR and educational resources, visit www.biorad.com/webinar. This URL is displayed to the left of your slide in the resources box. Thank you.